I can probably move this one. Okay, so here we see the frequency distribution of the heart rate variability, which is typically classified into three bands: very low frequency, and high frequency. So the high frequency is parasympathetic efferent vagal activity, whereas the low frequency is correlated to sympathetic and parasympathetic. So these are the parameters that we will extract from the heart rate variability, and the spectrum, the energy in the spectrum will typically drop down when the patient is less when the patient is less stressed. You will get a lower value. Can you move the next slide, please? Okay, so we were recording two EEG channels and one electrocardiogram with the Courses 1 monitor, which we see in the figure, sampling frequency on 1024 Hertz. So we have electrodes for monitoring EEG and for monitoring the electrocardiogram. Next slide, please. And here we can see the results. Here we were using two models for heart rate variability to define two indices. Either the eye nociception index based only on parasympathetic activity, and the second index based on a combination of sympathetic and parasympathetic activity. And we compared it to the QNOX index, which is the index I presented previously in the CONOX monitor. And we were calculating root mean square error between the heart rate variability indices versus the QNOX, and we were calculating MDAPE as well. There was not the two indices, the heart rate variability based indices were quite similar, as we can see here on this example here. There was definitely significant difference between the anesthetized state and when the patient recovered or versus awake. So this is an initial study in order to see if we can add information to a nociception index using heart rate variability, and I think definitely we can, because it is another modality which is important to include into the full description of nociception. So in conclusion, we calculated the two heart rate derived indices. They were not significantly different from the QNOX, which was calculated through the EEG. And we need to do more studies with larger population and different models in order to get to a final conclusion on whether adding heart rate variability to an EEG-based nociception index will provide a better prediction of the analgesic state of the patient. Thank you.